Welcome to another special episode of the Learning Report, hosted by Acquisition News. Today we will be featuring many guests to talk to you today about oxytocin and vasopressin, as well as the link between these hormones and autism spectrum disorder. To teach you a bit more about the topic at hand, here's our host, Stephanie Cherrycar.
Hello and welcome to another informative episode of the Learning Report. I'm Stephanie Cherrycar, and I will be talking to you about some basics that you will have to know before we speak to our guests. First, what are the hormones that are going to be the focus of our episode?
The first is oxytocin, a hormone that acts as a neuromodulator within the brain. It is responsible in social recognition, pair bonding, as well as maternal behaviors. The second is vasopressin, a neurohypophysical hormone that is important in social behavior, memory formation, and pair bonding. These two important hormones relate closely to autism spectrum disorder, which is a neurodevelopmental disorder characterized by impaired social interaction and communication that is associated with restricted and repetitive behavior. The individuals affected have such strengths as reading, motor skills, drawing skills, and are usually visual spatial learners. However, they have a variety of weaknesses that range from impairment in social relationships, dependence on routine, preservation on interests, and inadequate communication skills.
Some other information that will be useful in understanding today's episode is that oxytocin enables non-human animals to overcome natural avoidance. This hormone has been shown to inhibit aggressive behaviors. Oxytocin is released in response to positive social interactions. It has been found to promote social cognition and interpretation of social signals, possibly representing an enhanced readiness to show social approach behavior and empathy. It is also seen that vasopressin is responsible for male social behavior. We interviewed two scientists who do research on this very topic. Here's a clip of our interview with Dr. Vincent, who has conducted various experiments. We spoke to him about his research. So Dr. Vincent, tell us a little bit about your research.
Well, right now we're gaining supporting evidence that oxytocin modulates formation and maintenance of social bonds and regulates response to social isolation via its central receptors.
Are there any specific behaviors that you can tell us that oxytocin is implicated in?
Of course. The oxytocin pathways have been found to mediate forms of affiliation, including grooming, maternal, and reproductive behaviors.
Interesting. Would you like to tell us a bit about the research you've been doing in the lab?
Certainly. We use 6 to 8 day-old pups and house them with both parents and littermates. 45 minutes before the experimental procedure, the parents are removed and the pup is placed in an isolation chamber for 2 minutes. During this time, we measure the ultrasonic vocalizations, which provide us with a baseline measurement. After the 2 minutes are complete, the rat pups are injected with either peptide or oxytocin, which is then dissolved in the cerebrospinal fluid, and the pups are returned to their littermates. After another 20 minutes, the rat pup is released under its same conditions, and this gives us our experimental data.
So proceeding with your work in the lab, what did you predict the outcome to be?
Our original hypothesis was that the oxytocin receptors in the infant rat brains are functional, and the receptors will alter the expression of important signals in maternal retrieval.
What were the results of this experiment? Did they agree with your hypothesis?
Yes, actually they did. After the administration of oxytocin, we found that there was a significant decrease in ultrasonic vocalizations in the pups without affecting locator activity or coordination.
So what does this mean?
Basically means that when the oxytocin is injected, they don't feel socially isolated and don't cry for their mothers as much as they would without this extra oxytocin. With an increase of oxytocin, we see more social exposure and less social isolation. Also, the increase in oxytocin in infant brains changes the development of expression, which is related to the ability to socially learn.
What implications does this have for us as humans?
Even in humans, an increase in oxytocin could promote social behavior, which plays an important role in the formation of social bonds. If we were to release the oxytocin-injected individual into a social situation, we would expect to find the individual as in a state of social exposure rather than social separation.
Amazing. Thank you so much for your time. And now we will have a second experimenter who will be talking to us about his research with prairie voles and vasopressin. So tell us a little bit about your research.
Well, I do research with a monogamous species called prairie voles. Most species of mammals are polygamous, so this social behavior was unique and therefore was of interest to me.
So our viewers have had some background on vasopressin. Can you tell us a little bit about how vasopressin relates to these animals you're studying?
Well, we have found cells in certain parts of male voles' brains are loaded with protein receptors that bind chemically with vasopressin. When a male copulates with a female, vasopressin is produced and released into the bloodstream by other brain cells. It is then carried to the brain region that plays an important role in rewarding sensations associated with specific behaviors.
So what makes these voles want to stay with just one female?
When the vasopressin receptors are stimulated, a pathway is triggered, which gives positive feedback to the vole. This makes it want to stay with a specific female. The researchers believe that these rewards encourage voles to maintain a lengthy social bond with their female.
So is there something biologically different between voles and other polygamous animals?
Yes. Polygamous animals have far less VA1 receptors and do not receive positive feedback from one specific female, and so are not compelled to stay with her and often move on to the next.
Very interesting. And now we have two scientist parents whose son has autism spectrum disorder. Here they are talking about their experiences with oxytocin and vasopressin. Hello, I'm Dr. Smith, and this is my husband, Dr. Smith. And we're here to talk to you about ASD and our research on this disorder. After we found out that our son had it, it changed our lives, and we felt compelled to do research and gain knowledge in order to find out information about more therapeutic methods. 1 in 110 children are affected by ASD. New therapeutic approaches need to be discovered to make the lives of these children and their families easier.
Our first encounter with Vasopressin Oxytocin was made when there was research done to link the behavior of individuals with autism spectrum disorder to animals that lack oxytocin. The basic social effects of oxytocin and vasopressin we found may be applicable to human social behavior.
In one study, we saw that there was a lower plasma level in children with ASD, and there was found to be a correlation between plasma and oxytocin levels in social functioning.
In another study, it was found that oxytocin receptor and vasopressin receptor genes might even be involved in the development of autism spectrum disorder, and that a mutation can result in ASD-like behaviors.
It has also been shown in multiple animal studies that these same genes play a role in regulation of social behavior. This theory related to another proposed theory that the receptor genes play a role in autism.
After doing research, there is evidence showing oxytocin acceptor gene, region 3p25, as being a promising linkage site for ASD.
We have also seen that with systematic infusion of oxytocin injections, there is a reduction in repetitive behavior and improved recognition in ASD.
These injections could be a breakthrough and may become a significant target for novel therapeutic approaches.
Before we conclude, here's a message from our sponsors.
Does all this information interest you? Do you want to learn more about learning mechanisms and how it relates to our society today? Then PS261 is the course for you. Learn all about how you learn. Now, how does this course relate to the interesting information we have gathered here today? Social learning theory, covered in the course, states that people learn within a social context through such components as modeling and observational learning. As we have seen in the studies, a mutation in oxytocin and vasopressin in the receptors can alter social behavior. This alteration means that individuals will not be able to learn through the social learning theory. They would not be able to utilize classical and instrumental conditioning to learn social behaviors. For example, normally, a child would learn how to introduce themselves to a new person by observing their parents doing this multiple times. However, an individual with a mutation in oxytocin and vasopressin in their receptors would not learn in the same way because of their social impairments. This course on learning, please go to wlu.ca and look up PS261.
That concludes our segment for today. Thank you for watching, but before you go, let me leave you with some food for thought. The link with vasopressin and oxytocin with autism spectrum disorder is not for sure. As we have seen, there are many strong correlations as proven with the many research experiments with non-humans. If we can take the research that has been done, there can be efforts made to assist making the lives of people living with ASD easier. Again, thanks for watching.